Hello, everyone. Today I'll discuss how parasitic DNA, more familiar to us as transposable elements, can help shape our immune response. Our immune system allows us to recover from diseases caused by pathogens that have the potential to kill, initially providing a generalized innate immune attack and progressing to a specific adaptive attack. Our immune system is an integral part of our success as a species. The adaptive immune system is built on a key set of cells that orchestrate a complex sequence of events that ultimately keep us safe. T cells, short for thymus lymphocyte cells, are a type of immune cell. T cells work on the crucial principle of recognizing and discriminating between self and non self, ensuring to attack only the cells it identifies as non self. To reach this state, a learning process occurs during their development in the thymus. Up until recently, the mechanisms of this learning process were not well known, but an article in Medical Express discusses compelling research into the way it works. Researchers at the University of Montreal recently published a story in the journal eLife suggesting that transposable elements are responsible for facilitating the learning of T lymphocytes. Transposable elements, called genetic parasites in the article, make up 45% of our genome and, from my understanding of GEN2052, are often associated with disrupting genes and causing disease. For instance, the movement of transposable elements in the brain can lead to genomic alterations that cause Alzheimer's disease. However, this study demonstrates a different perspective. They uncovered that two cell types express high levels of transposable elements: medullary thymic epithelial cells, or MTECs, and plasmacytoid dendritic cells, or PDCs. Both cells are strongly involved in the immune response. MTECs produce and present self antigens, allowing for recognition and tolerance, while PDCs secrete interferons, which are signaling molecules that activate the immune response. After performing multi-omics analysis, their study revealed the methods by which transposable elements act. This article discusses this in a highly simplified manner for a non scientific audience, which is important. However, it left me feeling like I needed more information. By referring to the original research paper, I received a clearer understanding of the mechanisms by which transposable elements work. Transposable elements interact with transcription factors that are essential for MTEC development. For example, PAX1 and REL, and they generate MHC1 associated peptides, which are the receptors on cell surfaces that allow for recognition. They also discovered that PDC derived transposable elements form double stranded RNA that act to activate innate immune receptors. Cumulatively, these functions allow for T lymphocyte self/non self recognition. My interest in this article was sparked by the use case of transposable elements, as discussions in my Gen 2052 lectures indicate that transposable elements hurt us more than they help us, which I now know not to be entirely true. Though the author does not discuss it in the Medical Express article, they include an image from the original study which demonstrates both the various analyses performed as well as the different types of transposable elements present in the cells, which I could connect to the retrotransposon varieties we discussed in Gen 2052. The original study also dives into their research protocols and explains the reason behind choosing specific research methods. For instance, they utilize single cell RNA sequencing data to get a more comprehensive understanding of transposable element expression. Understanding the different techniques researchers use and the reasons behind their choice is important to me as a future researcher. This is the image from the scientific article. Finally, this article's tie-in to immunology demonstrates the interdisciplinary nature of genetics, which sparks interest to me as a double major student, hoping to combine genetics with other biomedical fields as a long-term research goal of mine. All in all, this article provided incredible insight into how transposable elements are integral to immune function. Thank you.